start with a little story that some of you may have even heard about. This guy that you see here in the picture is called Phineas Gage. He lived in the 19th century in the United States, and he was a railroad worker. He used to set up new tracks with his crew. And the way they did this was they'd go to an area which needed a new track, and they'd blow up the rocks so that they could make a new path for the railway. Yeah. Louder? Okay. So, and they use explosives in order to make a new path for the railroad. And unfortunately for Gage, this metal rod that you see here is the metal rod that he was using to stuff the explosives into the ground. And one of those days, the action of stuffing the explosives into the ground actually triggered an explosion, which ended up shooting this rod that he's holding straight through his cheek and out through the top of his head. It's kind of a gruesome story, I know, but the miraculous thing was that after he was blown away by the explosion, he got up. He was a little bit disoriented, but he walked himself to the hospital and he got treated. And within a couple of months, he was back on the job, and everything seemed fine. But what his family and his friends started noticing were changes in Phineas Gage's personality, which started to take place approximately after he came back to his job. He became very impulsive. He couldn't control his temper. He couldn't maintain his social relationships, and he ended up being isolated from his friends and his families, and he died in San Francisco about 15 years later from complications of lung disease. But the real story was that this was the first time in which the function of the part of the brain which was damaged by this metal rod came to light. And the part of the brain we know today, based on this computer reconstruction, his skull and the rod you can actually see in the museum in Harvard. And based on the reconstruction of the path of the rod through the brain, the scientists realized that the region of the brain that was destroyed by the metal rod is the region that we call the frontal cortex. Okay, this is the part of your brain which sits directly behind your forehead, and it's the part we know today which is responsible for all of our high-level cognitive functions. The ability to generate and understand language, the ability to retain items in short-term memory, so the fact that you can remember a phone number, like a 9-digit phone number, and then dial it, is really a result of the work of the prefrontal cortex, the regulation of emotions, regulation of social behavior. All of these highly complex functions are regulated by the prefrontal cortex, and this is the region that we study. Another reason why we believe that another reason we believe this region is so important is because in many cases of psychiatric disease, people have identified changes that take place in the prefrontal cortex, and these diseases include all these disorders that were already discussed in the movies that you saw: schizophrenia, autism, depression, addiction, ADHD. All of these different psychiatric disorders are associated with changes in the prefrontal cortex. But the problem is, and this is where we start, is that we have very little understanding of how these brain regions, these complex brain regions like the prefrontal cortex and the different structures of the brain that it talks to, how they do their job. We know that if we destroy a region, then we lose a particular function, but that doesn't necessarily mean that we understand how this region works, right? If I take away a transistor from the TV is going to stop working, but that doesn't really tell me anything about what the transistor is actually doing. So the problem really is the sheer complexity of the brain, and it's been already said that the brain is probably the most complex organ in our body. I think that's an understatement. It's really extremely complicated, and what I'm showing you here is an actual image of a very, very tiny part of a mouse brain. And what you see in these colored objects are individual neurons. So each one of these neurons is like a tiny computer. It gets its input, it listens to about 10,000 different neurons, and it somehow integrates all of this information and generates an output, which goes to an equal number of neighboring neurons. And how each one of these neurons works is already very, very complicated. If you think about each one of those neurons making 10,000 connections and trying to listen to all this information and make sense out of it, that's a very complicated task. However, so this basically leads to the current paradigm in neuroscience, which is that in order to be able to understand what these individual neurons are doing, we need to be able to control them. Because if we want to say that the yellow neurons control sleep, or the red neurons control aggression or social function, then we need to be able to, very much like in genetics, and the reason we have such great progress in the genetics of cancer, for example, is that in genetics we can take out a particular gene and learn about what that gene does, how that gene contributes to the function of the cell and to eventually to the disease. We need to be able to do the same thing for the brain. And over the years, people have tried to develop different ways of doing exactly this. And one of the really amazing discoveries has been that you can actually change the activity of the brain by using electrical stimulation. And I don't know how many of you have heard about deep brain stimulation. We were talking about it earlier today. So deep brain stimulation for Parkinson's disease is an incredibly effective tool for some patients. And it involves the implantation of tiny electrodes deep into the brain. So these are the electrodes. The electrodes are connected to a pacemaker, which is also implanted behind the shoulder blade. And the patients that suffer from very severe Parkinson's disease can undergo this surgery, which is a chronic surgery, and delivering electrical current through these electrodes actually leads to a really miraculous change in the symptoms, and some of these patients really gain back their life from this treatment. And this is because neurons are electrically excitable cells, and if you deliver electrical current, you can activate them or deactivate them in a particular way. And it's been suggested that this kind of treatment might actually be useful for other types of diseases of the brain, including depression and OCD and schizophrenia and autism. These are diseases that are not result of changes deep in the brain. These are diseases of the forebrain. These are diseases of the cortex, which is a very complex organ, as I just showed you. And unfortunately, these studies, which have been going on for the last 10 or 15 years, have been repeatedly failing. Okay, this is one item just from last year saying that another depression trial using deep brain stimulation for trying to treat the symptoms of depression has failed miserably, even though they had some initial very promising results in animal models. And we believe that one of the reasons that these trials are failing is because when you look at the cortex, you see the sheer complexity and the number of different types of neurons that are completely intermixed in the yellow neurons and the red neurons and the green neurons exactly in the same way. And if you do that in a circuit that has so many different functions mixed together, what you get is basically nonsense, right? You can't really achieve control of a specific function when you use electrical stimulation of a very complex brain area. So electrodes can't distinguish between neurons. So what can we do to obtain really selective stimulation of particular functions? Can we actually develop a method that can single out a particular population of neurons in the cortex and target those neurons selectively and only activate those neurons, and so that we can find the role of these neurons in the circuit? Not talk about brains, but talk about microbes. Okay, and what you see here is an algae. Okay, it's a unicellular algae. It's a very, very tiny microscopic organism which has very interesting properties. These are these algae, under the microscope. You need a microscope to see them. This is the algae in the dark. You can see them swimming around. They have tiny little motors and they can swim around. You can see that they swim around randomly. Let's see what happens when we shine light onto those algae. You see a change?
Yes.
Right, they start moving away from the light. That's really curious. And this is a mechanism that basically allows these algae to perform photosynthesis because this is their major source of energy. And about 15 years ago, it was discovered that these algae contain a single gene that allows them to do this. And I'll tell you more about this gene. So this is the algae. This is a region in the algae called the eye spot. This is what basically sees the light. So in the eye spot are tiny channels that when they're hit with blue light, they open up and they allow ions, electrical charged ions to go through. And this is what changes the behavior of the algae when it's illuminated by light. Now, the major discovery which took place exactly 10 years ago, just before I joined Stanford University, was that if you take the gene that codes, that is responsible for the expression of these channels, you take it from the algae and using genetic engineering, you introduce it into a neuron, then this is what happens. The neuron, almost miraculously, will start expressing those channels, those tiny opening channels on its membrane. And at this point, when we shine light onto this neuron, the neuron will become activated, just like the algae becomes activated, which allows it to move forward in response to light. And when the neuron becomes activated, it sends its signals, releases neurotransmitters to all of the neurons that it talks to. And in a way, this is mimicking the natural activity of the neuron, but this time we're mimicking it using light. Okay? So if we have a group of neurons in the tissue, and all of those neurons are expressing our gene from the algae, we can shine light onto the entire area. But now we're only activating those neurons that are labeled with the gene. Okay, we're really selectively addressing those individual neurons. And this has been a really fantastic discovery, and it's led to a complete paradigm shift in neuroscience. It's kind of become the way you do neuroscience because of the complexity of the brain. You really need a way to selectively affect the activity of individual cells, individual types of cells, so that you can understand something about these complex circuits. Okay, this is another color of light which allows us to inhibit the cells. So the gene that responds to yellow light is a different gene that's inhibitory. So we can use blue light to activate the neurons and yellow light to deactivate them. So kind of like a bidirectional switch. And even more interestingly, this actually works in living animals. So what I'm showing you here is an animal, a mouse, that has been engineered using genetic engineering to contain this gene in its brain. And this mouse is implanted with a tiny optical fiber, not an electrode. So all we're doing here is delivering light into the brain. And I want you to look at the video and tell me if you see anything changing in the behavior of the mouse when the laser starts illuminating the brain. With the mouse. So the region which was implanted with the optical fiber is called the motor cortex. This is the region of the cortex that controls movement, and specifically the region which is responsible for the hind limbs. And this is why the mouse started moving in response to light. Okay? Now, in the lab, I won't go into a lot of detail because I know it's late and you're probably tired, but I just wanted to tell you about the kind of questions that we're interested in our lab. So I told you about the prefrontal cortex. This is this region over here. The prefrontal cortex talks to the anterior cingulate cortex, and both of them are in very strong interaction with a very fascinating region called the amygdala. Has anybody heard of the amygdala here? Okay, the amygdala is thought of as the emotional part of the brain, right? It becomes activated when we're afraid, or when we're very happy, or when we're expecting something really good to happen or something really bad to happen. It basically serves as an alarm clock for the brain, saying, this is when you have to pay attention, something important is happening. And what we've been able to see is that the amygdala sends axons, connections into the prefrontal cortex, and using these connections, it can actually affect the functions, the cognitive functions that the prefrontal cortex performs. What we've been developing is a way to perform the same kind of deep brain stimulation that people do using electrodes to treat Parkinson's disease, but now we're using optical fibers instead of electrodes, and we can actually address very, very specific connections in the brain. So in one experiment that I'm showing you here, this is the amygdala of the mouse. It's connected to the prefrontal cortex of the mouse in exactly the same way that it is in the brains of each one of you. And what we've been able to develop is a method by which we implant a fiber optic, only activating those connections between the amygdala, and the prefrontal cortex. And by shining light in a particular pattern onto those connections, we can reduce the strength of the connection, and thereby rescue fear and anxiety-like behaviors in our mice. Another project that we're really interested in is looking at the generation of impulsive behaviors. And remember how I told you about Phineas Gage and how he became very impulsive? He started gambling, he couldn't control his temper. What we're interested in is understanding what are the cells in the prefrontal cortex, which neurons are actually involved in the regulation of impulsive behavior. And this is a video that I'll play now, but what I'll say just before that is that this is based on something like an iPad, which sits here at the end of this screen, and a region over here in which the mice can get sweet rewards. So the mouse learns over the course of a few weeks to perform very complicated cognitive tasks. If it's given this liquid reward, it's kind of like dulce de leche, diluted 1 to 10. It's very sweet, they really love it, and this is what it looks like. The movie is sped up 4 times, so you can see many different runs. So every time the light goes on, the mouse has to touch the screen very, very rapidly, otherwise it loses the reward. And the mice become really good at doing it. But what we find is that they're so anxious to get the reward that sometimes they touch the screen before the light appears. And this is what we use to understand how impulsive they are. And what we've been able to see is that when we silence, using our optogenetic tools, when we silence particular parts of the prefrontal cortex, the mice tend to do this many more times. They become more impulsive just by silencing a very specific population of neurons in the prefrontal cortex. And what we're doing now is trying to understand who these neurons are, what's their genetics, whether we can actually develop a method of activating them or inactivating them that would be generalizable to other kinds of diseases, for example, like OCD, which is associated with increased impulsivity and also addiction. And finally, I want to tell you about a method that we're using in order to understand autism. So about half of the lab is involved with studying autism and schizophrenia, and although you might think that these are completely different diseases, they actually share many features in common. And what has been proposed for these diseases is that they're associated with changes in the connections between the neurons. So I told you before that each neuron is connected to, on average, 10,000 different neurons. This is kind of like having 10,000 people in your Facebook network. You can imagine how complicated that might be. And what we're doing using our optogenetic tools is to record from individual neurons in the cortex. This is the prefrontal cortex of the mouse. This is an electrode that allows us to record from one neuron over here. And we can, using our very finely focused laser light, we can activate each and every one of those neurons in the neighborhood of this neuron, and we can basically figure out who are the neurons that talk to the neuron that we're recording from, and we can compare animals that are healthy and normal to animals that have mutations which cause autism and schizophrenia. And as you probably know, these are highly heritable disorders, so we can use the genetics of the disease in order to try to understand something about how the connections between the neurons changes in these diseases. So I'm just showing you here a video of one such experiment. So what my student has developed is a method that allows him to image. So this is using a very advanced microscope. He can look at all of the neurons in the 3-dimensional space, of the frontal cortex. And then once he has all of those neurons, he can label them and reconstruct the location of each one of those neurons. And once we have the location of each one of those neurons, we can address them, each one of them, with a focused laser and activate each one of those neurons separately. And then he goes through the neurons and stimulates them one by one while recording from this neuron that's labeled here in yellow. And what this gives us is basically a map of which neurons are connected, what's the strength of the connections, and we can correlate back this map of connections to the behavior of the mouse, and to try to understand what kind of changes in the connections between the neurons are associated with behaviors that are the hallmark of autism and schizophrenia, like social behavior, like cognitive functions, working memory. So, as I said, we're interested in how the network changes in psychiatric disease. We're also interested in the changes that take place during learning and memory. This is the part of our work that's not associated with disease, that just wants to understand how the brain functions in the normal state. So I think I'll summarize here in pictures. So we're interested in this network of brain regions associated with the prefrontal cortex, trying to understand how it works in the normal state and how it dysfunctions in disease. We use these genes from algae, we put them into neurons, and then we can address these neurons using light. We can excite them or inhibit them and try to understand how they work.
How do you put the DNA into the neurons?
Okay, so there are two ways to do this. One is generating a transgenic animal, and we have at Weizmann an excellent transgenic facility which allows us to make transgenic mice that contain these genes in their own genome. But an even easier way, which has been really fundamental in the lab, is we engineered viruses. If you know about adeno-associated viruses, these are viruses that all of us basically contain in our bodies. They're completely harmless, but we can actually use them as a method for inserting DNA into the cells that we're interested in. So the viruses are a really useful tool because they're very localized. You can put the virus in a particular brain region and it will go into the cells because that's what viruses do. But instead of putting its own genes into the cells, it's going to put the genes that we introduced into it. So this gives us the ability to go into very particular types of cells in particular brain regions. And then finally, we can correlate the physiological findings with behavioral findings and try to understand the links between the brain activity, brain connectivity, and behavior. Okay, this is my group. They're a group of extremely talented, gifted people. It's very interdisciplinary because of the nature of the work that we do. We have biophysicists, we have psychologists, we have biologists, chemists. And this kind of interdisciplinary effort is only possible, I think, in the Weizmann Institute, which really generates these collaborations and allows students from different faculties to go and do their research projects in the different labs. And I think this is one of the really powerful things about the Weizmann that I really like. So I'm done, and I'll be happy to answer any questions.